All right, this lecture is on Mendelian genetics, or as I like to call it, the physical manifestation of your genes. So, to give you an idea about how people used to think about things, like way back in the day, they used to think that when a child was conceived, there's this one parent and another parent, and they did this thing, and then all of a sudden, a baby was born. That's crazy, right? Like, bizarre. So they thought, because they noticed the babies or children's looked a lot like their parents, that there was some sort of weird blending thing that went on. They used to actually think that sperm cells had body parts of a baby, and egg cells had other body parts, and they came together, and inside the womb they actually fused and formed a whole organism. Which seems bizarre now, but back in the day that's what they used to think. So essentially they thought that there was this thing called the blending hypothesis, which is basically, if you take blue and yellow, you make green, or if you take white and red, you make pink. Okay? Now, this is not actually correct. Though it seems somewhat feasible, there are situations where it seems like traits blend, like in height of people, for example. Like two people of average height are more likely to have children that are also average height, but they also have the possibility that one of them will be really, really short and one will be really tall. Okay? We'll talk about a little bit about how that's possible in a bit. All right, so the alternative hypothesis was formulated by a monk named Gregor Mendel. Now, he worked with pea plants in a monastery, because what else are you going to do during that time, right? You pray, and then you study stuff. He tended the garden, so he started noticing these interesting things that were happening when he mated pea plants together. And his theory was called the particulate theory, or this idea that there's not really blending, but each parent contributes a distinct particle, as he called. And the traits being kind of the manifestation of the fact that you get one of the particles from mom, one from dad. And in some cases, some were dominant, the particles, and some were recessive for the trait. So, for example, if you have a purple and a white flower, if you mate a purple with a white flower, all the resulting flowers will be purple for the most part, depending on what particles that they had, right? And because of that, purple was dominant over white. It trumped it, okay? Another term for those particles, as we're going to talk about, is alleles. So the different versions of these genes are called alleles, but we'll get to that a little bit more in a bit. Just a quick refresher on molecular genetics and meiosis, okay? Just so we have a good frame of mind going into this. This is stuff we've talked about already. So we have 2 full recipe book libraries of DNA in each of our cells' nuclei, right? Those recipe books code for the proteins. You get one set from mom and one set from dad, and because you have 2 sets, you are diploid. The set we got from dad and the set we got from mom have the same recipes or genes in them, but they may have different versions of those recipes. Maybe some of the recipes were changed a little bit, different ingredients were put in there, okay? Those different versions were called alleles, or are called alleles, and because you have these different versions, they sometimes can cause differences in the dish that you make or the trait that you create in an organism. Okay, so remember, think of each individual homologous chromosome. Remember, you have 2 homologous chromosomes in each cell as one of the books in the collection of recipes. And then you have, like, you'd have a Chinese food recipe book that you got from dad, and a Chinese food recipe book you got from mom. And that is filled with different types of recipes in it, which would be representative of those genes, but your mom's recipe book may have slightly different versions of those same recipes, which would be like alleles compared to your dad. Okay? So remember, DNA replication is the process by which we copy DNA. We make a copy of the recipe book library to give a new cell that we're going to create through the process of mitosis, or in the case of some asexually reproducing organisms that don't do that, binary fission or something like that, you want to make 2 identical copies of the recipe book library so each new cell gets a copy so they can make all the proteins within those recipe books. When we want to make a protein from the recipe, we then copy that recipe in a temporary copy called mRNA through the process of transcription, and then it goes over to the chef. It leaves the nucleus. It goes to the chef, which is the ribosome. It gathers all the ingredients, which would be like the tRNAs that have amino acids attached to them, and adds them in order, which is the process called translation. And the result of translation is a protein, or in the case of this, the dish, right? The thing that we wanted to make. These proteins can do lots of different things in our cells. Some are enzymes, they drive chemical reactions. Some act as transport channels. Some as receptors to receive ligands and cause change within the cell. Transcription factors, structural proteins. There's all sorts of things that these proteins do. Now, the physical manifestation on the macro level, like the organismal level of all of these genes, is called a trait. All right, so some quick definitions. I'm going to run through these. You have to understand them because I'm going to use them as we're going through this, okay? So diploid, we should know this. You have two different versions or two different alleles of each gene because you're diploid, right? Our body cells are diploid. Haploid cells have only one allele for a gene. Our gametes, sperm and egg, are haploid. They are N. Remember, diploid is 2N. An allele is a different version of the gene. Remember, we just talked about that. Okay, so dominant alleles will trump or they're expressed over recessive alleles. So they tend to be shown more often than recessive alleles are. And if you have one dominant allele, it's always going to show over the recessive one. A recessive is only expressed if there's two copies of that recessive allele. So if you have, for example, a brown allele and a blue allele, you're going to see brown eyes because brown is dominant to blue. But if you have a blue allele and a blue allele, which are both recessive, you will express the blue eyes. And there's also this concept of being homozygous or heterozygous. So homozygous, the prefix homo means the same, and the prefix hetero means different. Homozygous means you have two of the same allele. So you can either be homozygous dominant, you could have two of the dominant alleles, or you can be homozygous recessive, where you have two of the recessive alleles. Heterozygous means you have one of each. So you have one dominant and one recessive allele. Okay? So now that we have those terms down, let's start talking about what Mendel found out. So Mendel had no idea what a gene or DNA was. His ideas just came from studying pea plants and looking at different ratios when he mated them together. So his experiments, and you have to know this lingo and jargon, his experiments consisted of 3 different generations that he did, a P generation, an F1 generation, and an F2 generation. It'll make more sense about how these all work together in a second when I kind of go through his experiment, but just be aware of that. So the first thing he did was he wanted to test plants that were pure breeding in his what's called the P, or the parental generation. That means they had 2 of the same allele for the trait that they were. So they take one purple flower that was pure breeding, meaning they would have 2 purple alleles, and then one white flower that had 2 white alleles in it. So that's the definition of pure breeding, 2 of the same alleles, so being homozygous for whatever trait. So when he bred the 2 together, he found that the next generation, which we're going to call the F1 generation, or the filial 1 generation, every single flower was purple. So he concluded that purple was dominant to white, because purple and white, when you cross them, you got all purple flowers. So as I said, P stands for parental, and the F1 generation, which was the result of the mating, so if you look here, so the result of the mating of the P generation individuals, those are all hybrids. They are all heterozygous. Now, he figured this out after the fact, but notice how all of the plants were purple in the F1. Now, if the blending hypothesis was right, you would expect that if you made it a purple and a purple, you'd get a purple, right? That just makes sense. Purple and purple. Okay, purple. Maybe the white flower trait disappeared. Who knows? But what he ended up getting was, in the second generation, the F2 generation, which was a result between crossing 2 F1s. So we'd cross an F1. Cross an F1 with an F1 individual. You'd get this weird 3-to-1 ratio of purple to white. So the white trait disappeared in the F1 and then reappeared again in the F2 when we mated the hybrid plants, the F1s, together. So you get this 3-to-1 ratio. Now, Mendel kept seeing this 3-to-1 ratio over and over and over again in lots of different traits. You can see here, over and over and over again, approximately a 3-to-1 ratio each time for all of these different traits. So he tried to make some sense of this. Okay? Now, we'll talk about some of the laws he came up with in a second, but we're going to start talking about how do we do these different types of problems. Now, the first thing you want to do, you always designate the dominant allele as a capital letter and a recessive allele as a lowercase letter. And we usually pick the capital letter for whatever the trait is that's dominant. So if you have purple allele is dominant over white, we're going to make purple big P. That means the white allele will be a little p. Or for eyes, brown's dominant to blue, so we're going to make big B, the brown allele, and little b, the blue allele. So after he did all of his experiments, he came up with 2 laws of inheritance based on looking at his data and logically thinking about, like, how is this all possible? The first law is the law of segregation. Now, a lot of students have difficulty differentiating between the 2 laws. Now, really, as long as you understand the 2 laws together, that's all you have to know. But I'll try to explain the difference between the 2 of them as we go through this. So the law of segregation is this idea that when sperm and egg are formed, each allele that you have, because you have 2, will be separated during the process of meiosis. And we saw that. That's the process of having a diploid cell and creating haploid gametes. We're separating the alleles apart through that process. So the traits are separate entities. They're not just one trait that you have. It actually has 2 components to it, which we'll call alleles, that the physical manifestation of those 2 components and the proteins being made from those alleles is what your trait is. The law of independent assortment, on the other hand, is this idea that during the formation of gametes, those alleles are randomly and independently assorted into those gametes. Not only are they separated, which is the law of segregation, they're also randomly, through the laws of probability, separated into the different sperm and egg. So if you have brown eyes and you have both a brown eye allele and a blue eye allele, or you're heterozygous, there's a 50-50 chance, because you're big B, little b, so I'll write that down. Big B, little b is your what's called the genotype, which we'll get to in a little bit. But your big B, little b, there's a 50% chance that if you're a male, your sperm will get the big B allele, and a 50% chance that they'll get the little b allele. So here's showing you the law of segregation right here, showing the 2, this is showing you a diploid cell, and these are 2 haploid cells. And you can see the gametes are separated into different, or the alleles are separated into different gametes, whether there be sperm or eggs. For the law of independent assortment, though, now this we kind of talked about, I think, a little bit when we got into meiosis, but it's this idea that now when the homologous chromosomes line up in the poles in the middle during metaphase 1 and metaphase 2 of meiosis, they're going to line up like this or like that. The way they line up is completely random. And what you can see here is we have 2 possibilities. You have the red, 2 reds here and here and here on the left side, and then 2 blues on the right side. But with just these 4 different chromosomes, we can also flip-flop it, right? We can get a red on this side and a blue on this side and a blue on this side and a red on this side. And as a result, from this process, you actually get 2 different types of gametes because of that. Now, if you were to actually look at all of the different possibilities, genetic possibilities that you can have, because we have 23 sets of these things, 23 pairs of homologous chromosomes, there's over 8 million possibilities just by doing the law of independent assortment and randomly assorting these chromosomes into gametes. Okay, so now gets the part that everyone loves or hates. I don't know. I'm kind of indifferent on them, but it's how to do a Punnett square. So, for example, we're going to do and look at Mendel's actual problems and how they relate to a Punnett square and how they relate kind of to this idea of independent assortment and probability and things like that. So his first cross was between a purple flower, which I'm going to put up here as big P, and then a homozygous recessive flower, which would be, or white, which is little p. Now, the way you do these is you put one of each type. It's kind of like factoring, right? So you put a big P and a little p here, a big P, little p, big P, little p, and big P, little p. So you end up with all individuals that are heterozygous, 1 dominant allele, 1 recessive allele. And because P, purple, is dominant to white, every single one of these is purple. So I'm going to write all purple, if you can even read that. Okay? Now the F1 that he did, the filial 1 cross, took the results of that parental cross and then mated them. Now every individual was big P, little p, so we're going to put big P, little p, crossed with a big P, little p. And now you factor, well, you got 2 big P's here, big P, big P, big P, little p, big P, little p, and then finally little p, little p. This results in a 3-to-1 dominant, because each of these shows the dominant trait because they all have big P's, to 1 recessive, or 3-to-1 ratio of purple to white. Okay? So that is kind of showing you why Mendel's results occurred using a Punnett square. Now, sometimes we look at Punnett squares with 2 traits. So the F1 cross we just did is called a monohybrid cross. It's called mono means 1 trait. Okay, mono is 1. Hybrid means you're crossing individuals that are heterozygous. But we can do things like dihybrid crosses too, and we can actually look at 2 traits. So for example, if we say purple is dominant to white, which we just did, but tall is also dominant to short. So we're going to cross a homozygous purple, homozygous tall plant with a white short plant. So the cross in this case would be right here. Homozygous purple, homozygous purple, homozygous tall, with homozygous white, homozygous short. The next step for doing these is to determine the possible gametes that are generated. Now remember, when you make a gamete, it has to have 1 of each trait only. And we do this by kind of like factoring and figuring out what are the possible gametes we can get. So we can only get 1p and 1t in each one. So in this individual right here, they can make only a big P and a big T gamete. Okay? This individual right here can make only a little p and a little t gamete. So those are the only gametes they can possibly make. So when we do this Punnett square, it's really simple. So this is like a 4x4. We're going to use this in a second, but we don't need any of these. The only one we need is this one square, because the only possibilities that we have are a big T, or big P, big T, and a little p, little t. And what results is an F2, or an F1 that is big P, little p, big T, little t. Now when you write these, you always write traits with each other, and you always write the dominant trait first. So every single one of the individuals, the only type of individual we can get from this cross is an individual that is heterozygous at both traits, or a purple tall plant that is heterozygous. But the F1 cross is a little bit more complicated than that. So now, what are the possible gametes we can get from these guys? So let's just look at the first individual right here. Well, we can get a big P, big T gamete, or we can get a little p, big T, or we can get a big P, little t, or a little t, or little p, little t. So we have 4 possible gametes. So each of these gametes is going to go in its appropriate spot on the Punnett square. So I'm going to do that. I'm going to draw them all in. Big P, big T, big P, little t, little p, big T, little p, little t. Now this is the same type of individual, so you're going to get the same type of gametes for them. Big P, big T, big P, little t, big T, little p, little t. Now you can actually go and do this whole entire cross, like you can factor them through. So like big P, whoops, big T, big T, big P, big T, little t, big P, little t, big T, big T. So you can see I'm going through each one and kind of factoring them all out, with this one being the only one that's little p, little t, little t. Okay, but you get lots of different ones throughout the whole thing. Now, if you were to do the whole thing yourself, you would get something that looks like this. Now, these are using R's, not P's and T's, but R's and Y's. But you get a very similar result. And what you end up seeing is this interesting ratio. You end up seeing, so if we circle all of the ones that are yellow and smooth, both of those being dominant traits in this case, you can count how many boxes have those traits. 1, 2, 3, 4, 5, 6, 7, 8, 9. So we have 9 with the dominant trait. How many show, or both dominant traits? How many show 1 dominant, 1 recessive? Well, the green's recessive, but smooth's dominant. So we'll say 1, 2, 3. So 3 show 1 dominant, 1 recessive. How about the other way? Okay, this is dominant for color, but recessive for like the shape of the seeds, which are wrinkly. So we have 3 recessive dominants, and we only have 1 that shows recessive recessive. All right, so as I said, this results in a 9 to 3 to 3 to 1 phenotypic ratio, where 9 out of those 16 boxes, it's a 4 by 4, so 4 times 4 is 16. 9/16 show both dominant traits, purple and tall. 3/16 would show dominant recessive, purple and short. 3/16 would show recessive dominant, white and tall. And only 1/16 would show white and short. And that would kind of be like your expected frequency if you were to have like 100 children. You know, you'd expect out of those 100, 9/16 of those 100, so multiply 100 times 9/16 to get the number of children you expect to have both dominant traits. You'd multiply by 1/16 times 100 to get the number you'd expect to have both recessive traits. And that is basically how inheritance works, at least simple Mendelian inheritance. There's obviously some more complicated things than that. So doing a 4 by 4 can be a pain. So here's a shortcut. Instead of doing a true 4 by 4, which takes forever to do, as you can see, I didn't even feel like writing it all out. You can also do two separate 2 by 2s with each individual trait on each separate Punnett square. So for example, we started with a big P, big T, little t, crossed with a big P, big T, little t. Remember, we have the same possible gametes here, but now we're just going to kind of separate them out. So we're going to only look at the P trait here and only the T trait on this one. So what are the possible gametes? Well, we can have a big P or a little p, or we can have a big P or a little p, and the same thing's true here, big T or a little t, big T or a little t. Those are the possible gametes we can get from each of these individuals, with this representing this individual, and then this representing that individual, and then this representing this individual, and this representing that individual at those traits. Okay, I'm going to erase those arrows to eliminate some confusion here. So now what we're going to do is we're going to do our Punnett squares. Now we should already know what we're going to get from this because we've already done this type of Punnett square before. And we get a 3 to 1 ratio in both cases, 3 to 1, dominant to recessive. So 3 out of 4 are dominant, 1 out of 4 recessive. Same thing here, 3 out of 4 dominant, 1 out of 4 recessive. So if you know the laws of probability, when you say and, that means you multiply the probabilities together. So what is the probability that you are going to have one that is dominant here and dominant here? We multiply the probabilities. 3/4 times 3/4, guess what you get? 9/16, right? What if we want the dominant and the recessive? 3/4 times 1/4, 3/16. How about this recessive and this dominant? 1/4 times 3/4, 3/16. How about the recessive times the recessive? 1/4 times 1/4, 1/16. So this is a shortcut. I think this is actually much easier to do, this method, than to do the whole dihybrid cross thing, where you do a 4 by 4, but to each their own. Okay, let's try another problem. So for the trait bean pod color, green is dominant to yellow. So we're going to basically call big G green and little g will be yellow. All right? And for pod shape, inflated is dominant to constricted. So we're going to call inflated big I and constricted little I. Please tell me the phenotypic ratio of the offspring in a cross between a homozygous green and heterozygous inflated individual with a heterozygous green and constricted individual. So first we have to write out what are the individual's genotypes, okay? So it says a homozygous green, big G, crossed with a heterozygous inflated, big I, little I, because it's heterozygous, okay? And then a heterozygous green and a constricted individual. Because constricted is recessive, we know it has to be little I, little I. Okay? Next step is to determine the possible gametes produced by each. Well, in this case, we can get a big G and a big I, or a big G and a little I. Those are our only possibilities. So these are our possible gametes we can get from that cross. Or not from that cross, from that individual. These are the sperm or egg that this individual can make. If you have big G, little I, right over here, big G, little I, little I. Well, you can have a big G, little I, or you can have a little g, little I. Those are our only possibilities here. Okay? So now we have our possible gametes. Now, even though this has two traits, this is still only a 2 by 2 Punnett square. So sometimes you can do it, and it's actually easier if you don't have a pure dihybrid cross to do a normal Punnett square. So here we have the same thing. We have our possible gametes for this individual. We'll put here. So big G, big I, big G, little I. And then this individual right here, we'll have big G, little I, and then little g, little I. And we do the cross, big G, big I, little I, big G, little I, little I, big G, big I, little I, and big G, little I, little I. So what we get out of this are individuals that have all big G's, right, at least one. So it's dominant in that trait, so it's going to be green. And then of those green individuals, half of them are inflated and half of them are constricted. So we actually have a 1-to-1 phenotypic ratio of green inflated to green constricted individuals. And it's 1-to-1 because it's a 50-50, right? Equal amounts of each. So if we were to do this cross and have 100, let's say we do this cross with flowers and 100 seeds are produced, out of those 100 seeds you'd expect them to germinate, you'd expect half of them to be green and inflated and half to be green and constricted. Okay. Same traits, different problem. So the trait for being pod color, green is dominant to yellow and pod shape is inflated and dominant to constricted. We just said that. Please tell me the phenotypic ratio of an offspring in a cross between a plant that is heterozygous for both traits, which we have right here, big G, big I, little I, and one that is yellow and constricted, little g, little I, little I. So for the first one, we have 4 possible gametes we can get, right? Big G, big I, little I. Well, we can have a big G and a big I, a big G and a little I, a little g and a big I, and a little g and a little I. So 4 possible gametes, which are represented here. For the little g, little I one, you can only get one gamete. You can get little g, little I. Remember? So this is showing diploid. This is haploid, remember. Okay? So because of this, you have 4 gametes on one and only one on the other. You can do a 4 by 1 Punnett square. That would look like this. Little g, little I, because that's the only possibility for the one, and the other one it's big G, big I, big G, little G, big I, and little g, little I, and then you kind of factor them together. Sorry, this looks bad, but big little g, little I, little I. So notice what you get out of this. Green inflated, so green inflated, a green constricted, a yellow inflated, and a yellow constricted. So this is a 1-to-1 to 1-to-1 phenotypic ratio. You get all of the different possibilities here. This is actually an example of something that looks very similar to what we're going to call a test cross, but we'll talk about that later. Okay, so 1-to-1 to 1-to-1 phenotypic ratio. All right, let's try another problem here. So an individual with the genotypes big B, or big B, little b has brown eyes and blue eyes is the recessive trait. That means you'll be little b if you have blue eyes. And also, if they are big R, or big R, little r, they'll have brown hair, and little r, they are blonde. If 2 people mate, the male has blue eyes and brown hair, just like this guy, who's Pierce Brosnan, mates with a female that has blue eyes and blonde hair. I don't know who that is, generic person on the internet. Is the male homozygous or heterozygous for brown hair if they have 2 children, one with blue eyes and blonde hair and one with blue eyes and brown hair? So the first thing we got to know, they both have blue eyes, so they both have to be little b. Or excuse me. Yes, little b, and she is also little b. Now we want to figure out, is he big R, little r, or is he big R, big R? We want to figure that out. She has to be little r, little b, because blonde's recessive and blue eyes are recessive, and that's what she has. But we're going to figure out what is he. So if they have one child with brown hair and one with blue, and one with blonde. So the way you would do this, all we have to do, because they're both little b, we know that they have to have blue eyes, and both children had blue eyes, so we don't have to worry about that. We have to look at, because we know what she is, so we're going to draw her right here. This is the classic female symbol, and this is the classic male symbol, so we're going to draw them here. We have the female being little r, but we want to know, is he big R, little r, or big R, big R? So let's test that first. All of the children, if you do this cross, have to have brown hair. There's no blonde option. Now, it said that one of the children had blonde hair. So we find out, and we know now, that he had to be heterozygous, creating a 50-50 chance of having a child that has brown hair and a 50% chance of a child having blonde hair. Okay? All right, so now we're going to add one layer of complexity onto this. We're going to look at a question that asks, well, what if it's a tri-hybrid cross? So you're looking at 3 traits. Each trait, both individuals are heterozygous at each locus. So it hurts my brain. You cannot do a Punnett square. It would be an 8x8 with 64 boxes. It would be way too much work. So in this case, we're just going to use that method I talked about before, which is where you take 3 separate Punnett squares, one for each trait, and then multiply the probabilities together. So we're trying to figure out, you're going to get a purple, short, green offspring. So that would means a dominant and a recessive and a dominant. What's the probability that you'd have that particular phenotypic result? So we know each individual is heterozygous, so we're going to draw that out here. Okay, and we can do each one, but each is going to be the same result. You get that 3-to-1 ratio, right? So I'm not going to bother. 3-to-1 phenotypic ratio. So it wanted dominant at A, so it wants 3 out of 4. That's the probability you'd have dominant at A. At B, it wanted a recessive, so 1 out of 4. And at C, it wanted a dominant, so 3 out of 4. So what's the probability you'd have this and this and this? You multiply the probabilities together, which ends up being 9 out of 64. And that's your probability. Okay? Another thing they sometimes ask you about is creating gametes. What's the probability you'll get a gamete given lots of traits? Now notice there's 4 traits in this case, A, B, C, and D. What's the probability we'd get gamete, big A, big B, little c, little d? Well, in this case, all you have to do is take each individual one. What's the probability we'd get a big A if your choice is big A or little a? Well, in that case, it's 1/2. Okay? What's the probability you'd get a big B out of a big B and a big B? It's the only option, so it's a 1. How about a little c out of a big C, little c? It's 1/2. And then a little d out of a little d, that's a 1. Okay? Multiply the probabilities, and you end up getting 1/4, or 25% of the time. Okay? And that's exactly the answer we get, 0.25. Let's try another one. Same individual, different possible gamete. So big A, big B, big C, little d, little d. What's the chance you get a gamete, big A, big B, big C, big D? Now you may start going through this, but you notice something right away. This is not possible right here, because we do not have a big D to give. We only have the little d's here. Okay? So this is not a possible gamete, so there's a 0% chance that that gamete would be created. Okay? All right, so now we're going to look at inheritance patterns that are different from like these normal Mendelian inheritance patterns, though they still follow kind of the same rules. They just kind of manifest themselves in different ways. So the first two we're going to look at are incomplete dominance and codominance. In incomplete dominance, it's kind of like blending. So if you have a white flower and a red flower, you would get a pink flower from that result. But it's not truly like blending, because if you take a pink and mate them together, like in the F1, the resulting F2 generation, you'll get a 1 to 2 to 1 ratio of red to pink to white. Okay, and we'll look at how that happens. in a bit. And the same thing, it's kind of similar with codominance. The only difference is both traits are expressed equally instead of blending. So where a white flower and a red flower would be mated to give you patchy red and white, and then the resulting F2 would be a red, a patchy red and white, a patchy red and white, and a white. Another example of codominance would be blood types, and we'll get into that in a second. So here's showing you incomplete dominance and what happens in that F1 generation and the resulting F2 that comes through that when you cross the F1s together. So you can see here we have a gene for color. R stands for red and W stands for white. And you take two pink individuals, so this one's pink and this one's pink, and you mate them together. Notice they're heterozygous, and when you mate them you get a red, two pinks, and a white. The red being the homozygous for big R, white being homozygous for W, and then the R and the W, and you're heterozygous, you have that pink color. In codominance, you end up getting this patchy situation. Here's showing you it in a chicken. So this chicken is speckled because it has both the black and the white genes to it. So if you have both, you're going to be speckled. If you have just the black, if you're homozygous for the black, you'll be black. If you're homozygous for the white, you'll be white. The common example that they cover, though, the more complicated example are the ABO blood types, the different types of blood that you can have. Now, everyone, I think it's common knowledge. You can be A, B, or O, but maybe you don't know why that is or how that happens. Now, this is an example of codominance, but also multiple alleles, because there's actually 3 different alleles that you can have for blood type, that at least that we focus on, okay? You can be big IA, okay? So big IA, which would be the A allele, big I, B, which would be the B allele, and then little I, which would represent O. And O blood means you have, basically you have no protein made. Okay, so that's more recessive. So the way this works is if you have a person that has A blood, they can be either of these two right here. You can be big IA, or big IA, little I. In the case of big IA and little I, big IA is dominant to little I, so A is expressed. So you have type A blood. B is a similar inheritance pattern there, except with the B's instead of the A's. If you have a big IA and a big IB, you're AB blood. But if you're little I, that's recessive. Now, each of these alleles codes for a protein that expresses itself on the surface of the red blood cells. So as you can see over here, we have type A blood, and this isn't what they actually look like, but it's, you know, a good representation of it, with these little kind of spikes coming off, and then the B proteins are like little circles, and if you're AB, you have both of those types of antigens on the surface, and if you're O, you have neither, because the little I allele codes for no protein at all. And you can do these crosses very similarly to the way that we did crosses for normal Mendelian inheritance. This shows you a homozygous dominant type A, homozygous dominant type B, and as a result, you can only get ABs from that. But we're still here on this other one, we still have type A crossed with the type B, but notice in this case, we get a completely different possibility in the offspring. You can get AB or B because this type A has a little I right there. Again, a different possibility. We get A, B, and A. And in this third possibility, you actually have the possibility to have O. Now, if someone has AB, it's not possible to have a child that has O because they have to either give an A or a B, so you're going to be at least one of those two types. So let's do a couple problems relating to blood typing. So a man with type AB blood, so we're going to make the male right here, and he's type AB blood, so big IA, big IB. Is married to a woman with type O blood. So she's little I. And she's a female, so we'll draw the symbol. The children's blood types are A, B, and O. They have 3 kids. Which child had to have been adopted? Well, let's see what are the possible. So one of the 3 children was adopted. So we have big IA, little I. So we have type A blood there, big IA, little IA, type A blood, big IB, little I, B blood, and, whoops, big IB, little I there as well. So what types do we have? We have B blood and A blood. We do not get O at all in this cross. So we know that the O child had to have been adopted. Let's try another one. Suppose a father has type B blood, so big I, B. Now we don't know what this is yet. We're going to actually try to figure that out. And a mother of type O blood, little I, little I. What are the chances that their next child will be blood type O? Now, let's see. And they have a child that is type O. So if he was type big IB, you couldn't have a type O child. So we know based on that this has to be, or this individual has to be big IB, little I. Then we do the cross. We get this and this. So what's the possibility of these that you'd have type B blood? 50% chance, a 1 out of 1, or a 50% chance. What are the chances that the child would have type B blood? So 50%, O, 50%, type A, 0%, and type AB, 0%, because those don't exist based on this cross. All right, another interesting way that genes can interact is through a process called epistasis. This is where one gene will actually affect the expression of another gene. So in this case, we're doing a kind of a dihybrid cross here between two individuals that are mice, obviously, and they have black fur, and they also have the second gene, which is a gene for expressing color, which is C. If you're big C, or big C, little c, you will express color. If you're little c, you will not. So when you do this cross, you would expect to get that 9:3 to 3:1 ratio, but you do not. In fact, you get a 9:3:4 ratio, because anytime you have a little c, which is in the case in these four, you're going to express no color. And it doesn't matter what you are here at all, you are always going to have white fur. Okay? So epistasis is when one gene affects the expression of another. Another type of inheritance pattern is called polygenic inheritance. And a lot of traits actually are inherited this way. It's when you have lots of genes that all affect one characteristic. For example, height or skin color or IQ. And a lot of these not only have multiple genes that are involved, but they also have environmental factors that affect them. But a lot of times something that has strictly polygenic inheritance, or that is dominantly inherited that way, shows kind of this bell-shaped curve of probabilities of having specific phenotypes, which a phenotype is what's expressed. A genotype are what the genes actually say. Okay, so you can see here a P generation, we have a very light individual, light-skinned, marrying someone that has very dark skin. And the F1 generation, you'd get all individuals that had intermediate skin. But if we mate the two intermediate skin people together, you get all of these possible gametes. Notice that. And you get this wide range of differences in the amount of skin tone darkness that you'd have. So for example, if you're dominant at all three of these loci, you'll have very dark skin. If you're recessive at all three, you'll be very light. And if you have different combinations of light and dark, you'll be somewhere in the middle. But this shows you that even two people who have average skin color would be likely to have mostly children that have pretty average types of skin color, as you can kind of see here. But they do have very small chances of having very light colored skin children and dark colored skin children on either side, which forms kind of that bell-shaped normal distribution. Pleiotropy is this idea that one gene actually has multiple phenotypic effects. So it's not just one trait that gene causes. It causes lots of different traits that sometimes are seemingly unrelated. Good examples of these are disease alleles that cause lots of problems within people, like sickle cell anemia, for example, which the normal hemoglobin gene is found right here on chromosome number 11. And if you have a mutated version of that, it can cause blindness, it can cause liver failure, it can cause lots of traits or lots of problems with the heart. And here it kind of shows all of these things as well: paralysis, impaired mental function, all because you have a hemoglobin gene that doesn't quite function properly. Cystic fibrosis does this as well too. It causes you to have this very thick mucus because you have this non-functional ion channel in your cell membrane that causes mucus that is produced by these cells to be very thick and viscous. Now, if that happens in your lungs, that thick, viscous mucus actually gets caught in there and accumulates and can cause scarring, which is where the fibrosis part of cystic fibrosis comes from. And cystic fibrosis will slowly kill you over the course of 20 to 30 years of your life. Most people don't live beyond 30 that have it. But cognitively, there's no issues there, so people kind of know that they're only going to live a very short life if they have it. But one of the weird things that you get with this, aside from, because this makes sense in terms of that, is you also tend to get clubbed fingers, which is another characteristic, completely seemingly unrelated to this disease. But in both cases, that's what happens. So, and notice it is found on chromosome 7, this gene. And it's just one tiny mistake in changing one amino acid, which is kind of crazy to think about. So, all right, so let's talk about single gene inheritance patterns. So if it's an autosomal dominant or autosomal recessive, there's kind of two different ways that we can do this here. An autosomal dominant disease is caused by having a disease allele where you only need one copy to have the disease. So it would be caused by a dominant allele. So if you were big A, little a, you would have the disease. The only time you do not have it is if you are little a, little a. And in autosomal dominant inheritance, one of the parents has to have the disease to pass it down. It's not masked in people like it would be an autosomal recessive, which you'll see in a second. So you usually see the disease in every single generation if it exists. It doesn't disappear and then come back again. Usually individuals, and this isn't always the case, but if you're big A, they usually do not survive to adulthood or reproductive age, or they're sterile. Autosomal recessive individuals, or I didn't mean that, an autosomal recessive disease is caused by having two copies of a disease allele. So in this case, they would be little a would be the disease. If you're big A, little a, you are a carrier, what's deemed a carrier for the disease. And if you're big A, you are completely phenotypically normal. Now, carriers usually don't express the disease at all. They don't seem to show any characteristics of the disease. But they do have this disease allele that can be passed on to their children. So this is showing you autosomal dominant inheritance. Here's an affected father. He can put either the big A or the little a in his sperm, and the mother is unaffected, so she can only give a little a or a little a. Notice if this is the case, you have a 50-50 shot of having a child that has the disease. Because you remember, only need one copy of the disease allele to have it, and that's autosomal dominant inheritance. An example of this is actually achondroplasia. So this isn't really a disease, it's just it's a kind of a condition which causes dwarfism. If you are big A, you have dwarfism. If you're little a, you do not. If you're big A, those individuals tend to die in utero or shortly thereafter. So if you have two people that have achondroplasia, which have a child, big A, crossed with big A, you would think like, oh, based on the blending hypothesis, that they would have to have children that also have it. But that is not the case. This individual would die, so they have a 1 in 4 chance of having a miscarriage. A 2/3, because we're not counting this anyone, 2 out of 3 chance of having a child that has achondroplasia, but a 1 out of 3 chance of having a child that is phenotypically normal, which is kind of interesting. In autosomal recessive inheritance, we have 2 carriers, a carrier mother and a carrier father, neither of which may even know, don't know that they have the trait, but you have a 1 in 4 chance of having a child that has the disease and a 50% chance of having a child that is a carrier for that disease. So this can be masked because they don't seem like they have the disease and they wouldn't even know, and then they get together, have a child, and have a 1 in 4 chance of having it. Cystic fibrosis is an example of this. This is a gene where it's an autosomally recessive inherited disease. It's super rare, but if 2 people that are carriers have children, they have a 1 in 4 chance of having a child that has it. And they're showing you that thick mucus in the lungs, trapped in the bronchial tubes of the lungs up there. All right, so related to this, newborn screening is a process of actually testing children for genetic defects and disorders at or before birth. Now, Jim Kelly, who is a former Buffalo Bills quarterback and is very active in this area and has had children that have gone to Narden, he created Hunter's Hope because his son had Krabbe's disease, which is autosomally recessive and an autosomal recessive inherited disease. So they were both carriers. So Jim was big A, little a, and his wife was big A, little a. And Hunter ended up getting Krabbe's disease. Now, for him, there was no real treatments or cures for it at this point, and there still is not. But he started this newborn screening thing to try to get children to, or people to get their children screened very early, because there are certain diseases that newborn screening can actually alter the case of and the progression of the disease, extending their life, making their lives better, and sometimes making it as if they never had the disease at all. So one way they do it is through blood testing and genetic analysis, but even before birth, they can do it through two methods, which are called amniocentesis and chorionic villi sampling, where in the amniocentesis, you'll actually take some of the amniotic fluid with some of the children's cells, the infants or the fetus's cells in it, and you would do genetic testing on those. Or you can actually earlier on test the chorion and the villi of the chorion and the cells which have some of the embryo's cells in them to test for these different diseases and disorders. So hopefully you can do some intervention to help them before damage actually occurs. All right, so let's kind of put all of this together and look at one particular disease called PKU, phenylketonuria. And that's an autosomal recessive metabolic disorder, and it's basically a mutation in a gene that normally breaks down phenylalanine. And phenylalanine is an amino acid. Notice the structure. Here's the R group, the amino group, and the acid group right there. Now, you'd think like, oh, well, why is it a big deal that you can't break it down? Because you just use it other places. So the enzyme is called phenylalanine hydroxylase that breaks it down. But if you do not have a functional version, phenylalanine will start to build up. And specifically, it builds up in your brain, in the blood-brain barrier. And that'll cause neurological damage, which we'll get to how that happens in a second. Now, these high levels are not due to what your body produces naturally through metabolic processes, because you can make phenylalanine. But what it does is if you eat phenylalanine, if you have a diet that's high in that amino acid, you get too much of it, and that's when the issue occurs. So if you get this diagnosed very early, and you fix their diets, they actually can live a relatively normal life. You can see here's showing that same inheritance pattern where you have a 1 in 4 chance of having the disease. This is actually the protein. Notice it shows, this is the quaternary structure here. There's 4 different subunits to it. Each of these subunits would show tertiary structure. If you zoomed in and looked at it, you'd see that alpha helices and beta sheets. And if you zoomed even closer, you'd see the individual amino acids, which would represent primary structure. Now, the way this works, why does it cause problems, is the protein that breaks this down is this enzyme, is the thing that doesn't work. So you accumulate this stuff. There's actually in the blood-brain barrier, to get phenylalanine and other large nonpolar amino acids into the brain, you have special transport proteins that do that. But if there's too much phenylalanine, it hogs the transport protein, and it clogs it up. And other individual amino acids cannot get through, and you need those amino acids to make proteins. So if this hogs it, other amino acids can't get across, and if they can't get across, you can't make every protein in your brain, which eventually would cause brain damage.